Hello, dear colleagues. We are now starting a session on multiple sclerosis and have a variety of topics around multiple sclerosis. And I think this will be a super interesting symposium. We will start the session with a young professor, Catherine Larochelle from Université Montréal. She is in the field quite a while and has done exciting work, and she will today have the teaching talk, the beginning of the symposium, with a completely new topic, which is, I think, extraordinarily important currently in the field, namely impact of biological sex and age on the course of multiple sclerosis. And please, Catherine, you have now the starting talk.
Good morning, everyone. So I would first like to thank the organizers and chair for giving me this great opportunity of a teaching talk on multiple sclerosis, and more specifically, I will focus on the impact of biological sex and age on the course of MS. So here are my disclosure, no conflict in relation to this presentation. And for the objective, after a short introduction, we will look at the impact of biological sex and age on first onset, then the course of MS, and then treatments of MS. So short reminder, multiple sclerosis is the most frequent demyelinating disease of the central nervous system, and it is characterized by lesions disseminated in time and space. And here you can see a typical MRI with typical lesions, and MRI is better suited to image white matter lesion. But if you look at brain, you will see that there are also some gray matter lesion here in the cortex and also in the white matter. And if you take a closer look, here you have luxulphase blue that would be staining myelin in blue. So you can see that the pathological hallmark of MS is this loss of myelin. And this happens in the vicinity of perivascular infiltrates by immune cells, such as T cells, B cells, and myeloid cells. And if you take a closer look, you will see that in lesion, but also in what would be called normal appearing tissue, there is glia activation and neuroaxonal injury. And therefore, we say that there are both neuroinflammatory and neurodegenerative components to multiple sclerosis. Now, we don't know yet what exactly happens to trigger MS, but we know that there are some genetic polymorphism that are considered risk factors that will interact with environmental risk factors and somehow lead to epigenetic and epitranscriptomic changes that will set things in motion and start some immune cell activation. And this may start multiple years before the actual clinical onset. And this phase of disease, called the prodrome of MS, is the subject of intense study and would be key to reach a point where we could prevent MS. And we also have lots of studies on radiologically isolated syndrome, where people have radiological evidence of typical MS lesions, but no clinical symptoms. However, in about 90% of the patients that will go on and develop MS, you will see an onset of relapse at some point. And these patients will then enter a phase called early MS, where they will experience clinical relapses and show a lot of inflammatory activity, formation of new lesions and lesions that will uptake gadolinium. And this phase is considered to be very inflammatory, with T-cell driven CNS targeted inflammation fueled by B cells acting as antigen presenting cells. And this is the phase where for the RRMS, we are really able to do something with the disease modifying therapy and to decrease significantly the relapses and the formation of new lesions. But then, unfortunately, over time, for the initially RRMS and in older individuals, in PPMS also, you will see onset of overt clinical progression or acceleration of progression, which might have started in started, in fact, much earlier. And this phase would be called late progressive MS. And in this phase, it's considered that the inflammation is mostly restricted to the CNS compartment. You'll have meningeal follicles, you'll have microglial activation, and on MRI, you will see mostly expansion of existing lesion and progression of atrophy, but much less highly inflammatory activity. And then we will say that this phase of MS is showing more neurodegenerative components than neuroinflammatory components. And in this phase, unfortunately, DMTs do not work as well. So at all of these stages of MS, there are factors that are influenced by biological sex and age. And this is what we will be looking at today. So first, if we look at MS onset, so it's pretty well known that as with many other autoimmune diseases, there is a female predominance in multiple sclerosis. And what is interesting is that before puberty, the sex ratio is 1:1. But after puberty, it's 3 women for 1 man who will develop MS. So one of the reasons for that might be, of course, sex hormone. And we know that these hormones will influence both the innate and the adaptive immunity. And estrogens are generally considered as immune enhancing, while testosterone and progesterone would be considered more immunosuppressive. What is quite interesting is that estrogen will kind of boost some of the T cell response, but will also be associated with higher antibody titers and maturation of the immunoglobulin. So globally, females or women are considered to mount a more robust immune response. But there's not only the question of hormone. In recent years, there has been very interesting studies published looking at the role of having 2 X chromosomes versus having just 1 X chromosome. So if you're a male, of course, you have only 1 X chromosome coming from your mother, while if you're a female, you have 1 coming from your mother and 1 from your father. So in female cells, 1 of the X chromosome will undergo random inactivation so that you don't double the dose of genes. However, there are some genes, so about 5% will escape this inactivation and will therefore be expressed to higher levels in cells of females than males. And some of these genes are implicated in regulating the expression of immune-related genes and will therefore influence the immune profile. And what is more is that if your X comes from your mother, there will be less methylation of some genes, for example, FOXP3, which is really important in regulatory T cells. So you will express more FOXP3 if the X that is active is from your mother, which is always the case for males, of course. But if the X that is active is from your father, these genes will be more methylated and will be expressed to lower levels. So this again could push a bit the immune profile towards a pro-inflammatory immune profile. So this is an obvious example with the X genes and the female hormones. But what is also important is that you have an interaction of biological sex with some of the environmental risk factors for MS. And the first one I want to talk about is obesity. So in the last decade, it was shown by multiple studies that obesity before 30 years old and more likely even more in kids will increase your risk of MS. And in fact, if you have genetic risk factors for a high BMI, you will also have a higher risk of multiple sclerosis. And what is relatively well known is that obesity is increasing in kids in recent years. But what is maybe not as well known is that, in fact, boys are more often obese than girls. So there's a difference in relation to sex. But after that, in adulthood, women are more often obese than men. And there's multiple studies showing that sex and gender have tremendous impact on dietary habits, on exercise patterns, and on multiple lifestyle choices that might be influencing BMI and also the risk of MS. So this is already interesting, but what is more also is that obesity has been found to be associated with more severe optic neuritis only in males. So you can see that there's no association with BMI with the severity of optic neuritis in female, but there is in men. And also there's been described that leptin, which is a hormone that is considered pro-inflammatory and is higher in women and in obese individuals. So higher leptin levels are seen in kids with multiple sclerosis compared to healthy controls. And the difference is more important when looking at boys with multiple sclerosis compared to age-matched healthy controls. So this outlines the fact that there are complex interaction between biological sex and environmental factors in MS. And another environmental factor that is important, so you most likely have heard about this publication in Science, showing that in young adults who were initially seronegative for ABV, the ones that will seroconvert, so will have somehow been exposed to Epstein-Barr virus or have developed infectious mononucleosis. So those who will become seropositive or are at much higher risk to develop multiple sclerosis than the ones who would have remained EBV negative. So this seems to show that EBV infection precedes the onset of MS and puts you at risk of developing MS. And the link with age and biological sex is that first, the risk of contracting EBV increases with age. And in developed countries, there is a lot of infection happening in teenage years. And what is also interesting is that there is a higher percentage of seropositivity among girls and women as compared to men. So if you look at the biological mechanism that could be implicated in how ABV promotes the onset of MS, so there's been multiple studies, but a recent one has suggested that there is some molecular mimicry between ABV-related proteins and cell proteins that would be expressed in the CNS. And in MS patients, you can see that the reaction to the EBV protein is higher, and to some specific peptides that would be very similar to our self protein would be higher in MS than control, so suggesting that higher titers of antibodies would be associated with multiple sclerosis. And this was done mostly in women. But what is interesting and might be an avenue for future research is that antigen response, as we said previously, are generally more robust in women, and higher ABV antibody titers are found in girls and women. So this might also be a way how biological sex would influence the impact of an environmental factor in MS. So this was for MS onset, but now if we look at the course of MS and the impact, for example, on relapses. So first, of course, as you probably know, in the relapsing remitting form of MS, you have 3 women for 1 man, while in primary progressive MS, where you have almost no relapses, you have 1 woman from 1 man. So outside of the factor of showing relapses or not, what about the number of relapses? So if you look at the sex ratio, comparing the individuals with MS showing a low relapse rate or a high relapse rate, what you see is that the sex ratio from woman to men will increase. So you have 3 women for 1 man in the people showing a high relapse rate. Well, it's more likely 2 to 1 in the one showing lower relapse rate. So it means that women show more relapses. However, on the contrary, they show a better recovery following relapses. So risk factors associated with incomplete recovery from relapses are prior disability, older age, and male sex. So this is also translated in the disability accumulation over years. So if you look in our RMS, you can see in blue that men have a consistently higher mean EDSS for the same disease duration, while in PPMS there is no such difference. So this brings the question, how do patients accumulate disability in MS? And this is a very recent article that is really interesting and that shows that both relapses, where you have incomplete recovery, and progression independent of relapse activity will contribute to the accumulation of disability, even in progressive MS. And among the most important risk factor, there was pre-existing disability, but also male gender and older age, that are the major risk factor for incomplete relapse recovery, and also for progressive MS and progression in MS. What is also implicated in progression and will result in accelerated progression are cardiovascular comorbidities. And of course, these will increase with age. And it has been shown that, in fact, for example, the Framingham risk score, which is a score of your cardiovascular risk, is associated with higher disability in multiple sclerosis. And in fact, cardiovascular comorbidities are also associated with higher rates of atrophy, higher risks of relapse and disability progression. And if we look at the prevalence of cardiovascular comorbidities, you can see that most are more prevalent in men in blue than in women in red. And you have a higher prevalence of hypertension, diabetes, and heart disease in MS than in healthy control. And you can see that, again, there are some differences according to biological sex, and the highest prevalence is in men with MS. And of course, you will have an influence of age also on the occurrence of these cardiovascular comorbidities. And again, if we look at the impact of age in MS, it's pretty obvious. As we previously stated, aging is the main risk factor for conversion to a progressive course of MS. So at onset, you will have virtually no primary progressive MS in the pediatric population, while in the late onset MS, so onset after 40 or 50 years old, you can have up to 40% of the individual who will start as primary progressive MS. And we know that late onset MS is associated with a shorter time to reach the SS of 6 or to convert to a secondary progressive course. And also mean age around the onset of progression, as we said before, is similar between progressive, secondary progressive and primary progressive MS. So what is also of concern is that not only is aging associated with disability and comorbidities, but it's also associated with a lower efficacy of our disease-modifying therapy. So apparently the benefit from treatment decreases with aging. And what is, of course, even more concerning is that the risk of adverse effects, so the risk of opportunistic infections such as vis-a-vis reactivation or PML, the risk of arterial hypertension secondary to the use of our DMTs, the risk of malignancies and low immunoglobulin are all increasing with aging. So the benefit to risk ratio is likely decreasing with age. And there's a point, of course, where we will need to decide how and when to stop the DMTs, and this is the subject of intense research. So what could explain this decrease efficacy of our treatment and the rise of the infectious and neoplastic risk? So this could be the biological aging of immune cells, which is also called immunosenescence. So with aging, what we see is that globally there is a lower capacity to mount a good immune response. So if you vaccinate older individuals, and we've seen it a lot with COVID, so vaccination is not as efficient in older individuals. And globally, you have a reduction in thymic output, you have less naive lymphocytes, you have less proliferation and renewal of your cells. They show lower phagocytosis and secretion of trophic factors. So globally, you have this lower capacity to mount a good response. But on the contrary, you have a chronic low-grade inflammation, you have more memory lymphocytes, you have more oxidative stress, you accumulate DNA damage, there are elevation of some pro-inflammatory cytokines like IL-6, you have higher levels of CRP, more autoantibodies, and globally, the aging of the immune system is associated with what is called inflammaging, so a chronic low-grade inflammation. So if we look at the biological mechanism, there are many different ones. So you have thymic involution, this pro-inflammatory environment, and you can see the telomere shortening, which is one of the hallmark of biological aging. And globally, you have some kind of dysfunction, tissue disorganization, and this is associated with dysfunction of the immune response. And what is important in MS is that repeated antigen encounter will accelerate the aging of the immune system, as will elevated systematic oxidative stress and chronic inflammation, because this will increase cellular turnover, and then you are at risk of replicative senescence. And we indeed see in autoimmune diseases that there are elevated numbers of age-associated B cells that are enriched in autoantibodies, and there is also elevated frequency of CD28 negative T cells. So these are T cells that show cytotoxic properties and are considered as older cells, if you will. And we do have proof from multiple studies that there is indeed premature immunosenescence in multiple sclerosis. So even in pediatric MS, you can see that in kids with MS, there are lower proportion of naive T cells compared to age-matched control. And in adult MS, there's been multiple studies showing that there are shorter telomere length in MS compared to control. And what is particularly interesting is that a shorter telomere length is associated with higher disability progression. So how does it happen? Well, immunosenescence is not only a problem for mounting a robust immune response, it's also a problem for neurodegenerative processes. So there's been studies showing that, in fact, inflammation is associated with neurodegeneration. There is an increased frequency of pro-inflammatory Th17 cells in Parkinson's disease. But also, if you look at young immune factors, they will restore the capacity for CNS repair. But aging, for example, macrophages and microglia will be associated with inflammation, but with a lower capacity to clear debris and secrete trophic factors that would be necessary for repair of myelin and axonal growth and maintenance. So there are a lot now of anti-aging interventions that are tested in MS and its animal model, for example, metformin, fasting, low methionine diet, and there are more to come to try to somehow address this this phenomenon of immunosenescence as a driver of neurodegenerative processes. And if we come back to the biological sex, there are very complex interaction between biological sex and biological aging. And if we want to progress towards a really individualized precision medicine, this will have to be taken into account, even if it's extremely complex. So you all know that the lifespan for women is generally a bit longer than for men. And the process of biological aging is different in the different organs according to biological sex. And you have these different conditions that are more frequent in men with aging. You have others that are more frequent in women with aging, and some that show very complex relationship. For example, frailty, which I think is an important field right now in MS. You have higher levels of frailty in women, but frailty is associated with a greater risk of mortality in men at any given level of frailty. So these are really complex interaction and will be of tremendous importance for future studies in MS. And I hope that today I have convinced you that biological sex and age are very important factors to take into consideration at all stages of multiple sclerosis. And with that, I'd like to thank you for your attention, and we'll be ready for any questions. Thank you.
Yeah, thank you very much, Catherine. This was really a wonderful overview on really very relevant factors for multiple sclerosis, but also brain diseases and autoimmunity. So if we want to go a little bit into depth in a few points, I would like to start with the question, if you look at inflammaging as a chronic pro-inflammatory condition, why do the MS patients not have rather increased numbers of relapses and guard enhancement during age? What do you think? What is the question? And is inflammation really within the brain in MS dying out over time, or is that a wrong perspective?
Well, that's a really interesting and very complex question. So first, for the idea that why aren't older people having more relapses? I think that the aging of the immune system is very complex because it's pro-inflammation in some ways, like with the increase of IL-6 or CRP, but it's also a bit like burnout, the robustness of the response to antigen triggers, for example, like with vaccination, is not as robust as in younger people. So what I would speculate is that in older people, you don't have such a flare, such a big rapid response of the immune system when you have a trigger, for example, environmental trigger, but that you have this kind of low-grade chronic inflammation that is somehow contributing to the fact that you lose axons, you lose your myelin, you have kind of a toxic environment for your CNS, and then the CNS is not able to repair as well, but you don't have this kind of spectacular flares of inflammation like you would see in younger people.
Okay, here are many questions now from the audience all of a sudden, and I hope that we will be able to answer them all. A few. You got a lot of comments on an excellent talk, and now one question. You mentioned that as individuals get older, patients with relapses switch to progressive MS. Do females make this switch more often than males?
That's a super interesting question also, and I would say that it's hard to give you a perfect answer, but from what we know is that because you have just more women with RRMS, you're going to have more women with SPMS. But if you look at RRMS in females versus males, you're going to have a shorter time to conversion to progression in males usually than in females. So usually male is the biological sex of male is considered to be a risk factor for the onset of progression. So you would say that mostly our benign MS that we will see after 30 years, they are still RRMS, they have not progressed. Usually they are females.
And another question, in many MS articles looking at effects of age on treatment responsiveness, they often use less than 40 years or more than 40 years. Why is 40 years the threshold where treatment responsiveness is much lower, or is 40 years arbitrary?
Well, I think it's a bit of both. I think it's a bit of a, usually in most studies, they include people younger than 55 or 60 years old, just because they want to have the best benefit to risk ratio. So they don't want to have older people who might have cancers or opportunistic infection. But also 40 years old, if you look at it from a biological point of view, that's the moment also where you will see fertility decrease, especially in women. And you start to see also androgen drops also in men. So there's also kind of a real point where you can say that 40 years old would be such of a sort of a threshold. And also when you look at onset of progression, both in PPMS and SPMS, this would be between 40 to 50 years old in most individuals. So yeah, I think it's both arbitrary and somehow a bit based on biology.
Yeah, we will not be able, unfortunately, to answer all the questions. However, I encourage, nevertheless, the audience to ask many questions, already very interesting questions. Maybe one last for this talk now. Could you elucidate on pregnancy as a factor? You didn't specifically come to pregnancy.
Yeah, and it's super important. It was just a question of time. So during pregnancy, especially during the second and the third trimester, there is a clear decrease of relapses. And after giving birth, there might be some kind of a surge of relapses, or or some people say that it's like the relapses you would have experienced during your pregnancy are all happening in the first trimester postpartum. But yes, there is really a protective effect of the pregnancy hormones. And it has been tried to replicate it with the hormones that you give, synthetic hormones. And so far, it hasn't been as successful as we would have liked. But yes, certainly pregnancy is kind of protective and almost like it's as good as most of the treatments.
Yeah, thank you very much, Catherine. This was wonderful for the start. Thanks a lot. And we now come to a data blitz talk by Alberto Calvi. He's coming from UCL London, Queen Square, and his topic is now, I think, fitting very well, slowly expanding lesions associated to persisting black holes and clinical outcomes in relapsing onset multiple sclerosis. Please, Alberto.
Thank you to the organizers for selecting this work for the multiple sclerosis session of the conference. This MRI analysis, carried out at UCL Queen Square MS Center as part of my PhD, I received the support of the Guarantors of Brain, the UK MS Society, Magnims and Ectrims, and the work was realized with the partnership of University of Siena and Milan. Chronic inflammation, neuroaxonal loss within white matter lesions are key contributing factors for disability progression in MS. Slowly expanding lesions or cells have been described in MS trials as a marker of chronic inflammation. They are extracted from the baseline T2 lesion mass through registration of the follow-up MRI scan and the formation field analysis. These initially identify candidate cells from the positive expansion as opposed to non-cell, and then subselect the fraction of lesion with constant and concentric features of expansion, the definite cells. As a surrogate of structural damage, the persisting black holes, or PBH, are lesions with chronically low T1 signal. In this work, we developed a pipeline based on selecting lesions by their mean T1 value below a threshold based on the gray matter T1 intensity, occupying at least 10% of the individual lesion volume. The objectives of this work were to assess the relationship between cells and PBH as a retrospective MRI analysis of 135 relapse-onset MS patients over a 7-year follow-up, evaluate their structural damage by computing lesion level T1 and magnetization transfer ratio, MTR, and finally to identify their contribution to clinical disability. Out of the total T2 lesion count, nearly 1/3 represented cell in this core, and among the total black holes, the PBH were more than half. From the table, at the lesion level, higher percentages of both possible and definite cell were also PBH. And on the plot, the T1 value over time had a significant decrease in these categories compared to the non-cell. In keeping with this result, MTR cross-sectionally was also lower within cells. Here, an example of a patient in this cohort with a lesion that corresponds to both a PBH and a cell. From the clinical perspective, in mixed regressions, we found an association between an increase of the DSS score, which relates to higher MS disability and the cell volume, and higher risk of confirmed disability progression in the logistic model. Thank you for your attention, and I would try to answer to all your questions.
Yeah, thank you very much, Alberto. Wonderful. Let me start with the first question. Could you relate your data to smoldering inflammation, the very new concept and very interesting and I think conceivable concept in our field, which may have to do with expanding lesions?
Yeah, thank you for the question. Professor Zipp is very interesting, the topic of smoldering inflammation. I don't think there is an unique answer, but what I can say about this work is that slowly expanding lesions are one of the markers of this smoldering inflammation, as they try to capture this slow expansion, which sometimes cannot be really found in especially in those patients that are in the progressive phase, as also Catherine was speaking before. So I think this is one of the possibilities. There are many other markers, but I think this is also interesting because it could be automatically extracted, so might be facilitating also in clinical practice the how to measure this smoldering inflammation.
And concerning your relation you found, was this your hypothesis, or were you surprised with your findings?
Yeah, thank you again. So yeah, I think we were sort of expecting also this result, because in general we think also that there is an accumulation of neuroaxonal damage in those type of lesions. So they are characterized by slow inflammation. So in the periphery, these kind of chronic microglia areas that are still functioning and burning in a way, but there is also a component of neuroaxonal loss, so this damage that here we were measuring with this other way, so the black holes, so this hypointensity in the lesion, so they are associated in a way. We found that, yes.
Okay, so I also encourage my colleagues from this session who speak here to ask questions. But if there are no more questions from my colleagues and from the audience, we thank Alberto once more. Thank you very much for this wonderful Data Blitz talk. And we now come to the next bigger talk given by Nathalie Arbour from also Université Montréal, from the CHUM. Nathalie is talking on the basic science concerning contribution of IL-27 and NKG2D to neuroimmune responses in multiple sclerosis. Please, Nathalie, it's your talk now.
Hello, everybody. It's a great pleasure today to present my work. First of all, I would like to thank the organizers for giving me this opportunity. And I am working at the CHUM, which is in Montreal, in Canada. So this is my disclosure slide. So the overall hypothesis of my research program is that immune mediators that are altered in MS patients modulate and shape T-cell responses and activation. And our long-term goal is to identify and characterize immune mechanisms that contribute to the neuroinflammation we observe in MS. And we want to look at both what's happening in the periphery, but also what's happening in the central nervous system of patients. The first molecule I would like to talk to you about is the results we obtained for IL-27. This cytokine is composed of two chains, P28 and EBI3. And this cytokine is produced upon activation of different myeloid cells and antigen-presenting cells. The receptor is composed of GP130, which is shared with other cytokines, and IL-27 receptor alpha chain. The receptor is expressed by multiple immune cell subsets, and I would like to take your attention to the CD8 and CD4 T cell subsets. So the first thing we did is we look whether or not IL-27 is expressed in the postmortem tissue of MS patients. So Vincent Sénécal, a previous graduate student, looked in postmortem brain tissues, and he identified cells that were co-expressing the two subunits of IL-27, so P28 and EBI3. And he quantified those cells per surface area, and he realized that the number of cells that were double positive in MS patients was much greater than what we could assess in control tissue. Later on, Marie-Laure Clénet also assessed the levels of IL-27 in biological fluids from MS patients, and she observed that in both in the serum, but also in the cerebrospinal fluid, the levels of IL-27 were higher in MS patients compared to healthy age and sex matched controls or compared to other neurological diseases. The other observation that was also important for our work is that within the postmortem brain tissues of MS patients, we can identify subsets of CD4 T cells and CD8 T cells that express the IL-27 receptor. So therefore, T cells from MS patients that have infiltrated the brain of those patients can also respond to IL-27. So the question we ask is, what is the impact of IL-27 on T cells from MS patients? IL-27 is known that upon binding to its receptor is triggering the phosphorylation of STAT1 and STAT3. And the different human T cell subsets were characterized as being naive, central memory, and effector memory based on markers that most of researchers are using. So what Marie-Laure did is she looked at what's happening in T cells from MS patients and healthy donors regarding the phosphorylation of STAT1 and STAT3. What she observed is that in T cells obtained from healthy donors, illustrated here by the white bars, and in naive central memory and effector memory subsets within the CD4 compartment, but also within the CD8 T cell compartment, each dot represents an individual donor, and you have in the tinted bars the results obtained from the MS patients. And as you can see, the percentage of cells expressing STAT1 phosphorylated after IL-27 exposure were very similar between healthy donors and MS patients for both T cell compartments. However, when she looked for the phosphorylation of STAT3, she observed a significantly greater percentage of CD4 T cells and CD8 T cells, and that was true for all the subsets we have looked at, that those cells were greater percentages in MS patients compared to healthy donors. And I have to mention those experiments were done on the same cells. So definitely IL-27 triggers greater STAT3 response in T cells from MS patients. We were also intrigued by the observation reported in Crohn's disease patients where the receptor of IL-27 is found as a soluble form in the serum of those patients in elevated amounts, and that such soluble receptor alpha serves as an antagonist for IL-27, preventing IL-27 from acting on the IL-27 receptor on the surface of target cells. So Marie-Laure assessed the level of the soluble IL-27 receptor alpha chain in MS patients, and she observed a greater amount compared to what she could see in healthy donors that were age and sex matched. She then assessed whether or not such level of IL-27 receptor alpha soluble could also have biological impact. So she used the assay that we have, which is to assess STAT3 phosphorylation in response to IL-27. As you can see, CD4 and CD8 T cells triggered nice response to IL-27. However, as she was adding in the same time increasing amount of the receptor soluble in the assay, she could block the capacity of IL-27 to trigger signaling in both CD4 and CD8 T cells. I would like to draw your attention that the dose at which we reach significant, I would say, impairment of IL-27 signaling was around 30 nanogram per ml. And this is about the median, I would say, amount of IL-27 receptor alpha chain we measured in the serum. So to summarize this part, I showed you that IL-27 levels are elevated in the brain, but also in the cerebrospinal fluid of MS patients. IL-27 is also elevated in the periphery of MS patients. I've shown you that T cells can respond to IL-27. STAT1 phosphorylation is about similarly induced in T cells from MS and healthy donors. However, STAT3 phosphorylation is induced at greater levels in T cells from MS patients in response to IL-27. I did not have time to show you today, but the IL-27 is able to decrease the production of IL-17 and GM-CSF by activated T cells, and this capacity of IL-27 to downregulate those cytokines was very similar between MS patients and healthy donors. However, the capacity of IL-27 to increase the expression of PD-L1, which is an inhibitory ligand that T cells can express. This capacity to trigger this, I would say, inhibitory ligand was reduced in MS patients. We also, I also showed you today that the level of soluble IL-27 receptor alpha chain were higher in the serum of MS patients. And I did not have time to show you today, but we have data showing that activated CD4 T cells are at least responsible for shedding this higher level of IL-27 receptor alpha in MS patients versus healthy donors. And those levels are sufficient to block the biological impact of IL-27, as I have shown you in the last few slides. So the second molecule I would like to talk to you about is the NKG2D pathway. So healthy cells, when they are exposed to different stress, such as inflammation, ER stress, are going to trigger some ligands, which makes them seen as abnormal cells by different immune cells. So upon these stress, if the cells start to express the NKG2D ligand, they can be recognized by immune effector cells that express NKG2D. And upon such recognition, these immune effector cells are going to increase cytotoxicity, lytic enzyme release, and production of pro-inflammatory cytokines. In humans and in mouse, these ligands are coming in different flavors. So we have different ligands that have been identified in humans, as they are illustrated here, as well as in mouse. What is important to remember is that the stimuli or stress that is going to trigger each of those different NKG2D ligand has been shown to be context and cell type specific. So that's why it is important to assess whether or not specific stress conditions can trigger specific ligands. And then the first thing we did is we assessed whether or not the NKG2D pathway has an impact in vivo. So a previous graduate student in the lab, Lorraine Legros, she induced passive VAE, and she compared wild type and NKG2D deficient recipient. As you can see, those mice, upon receiving activated MOG-specific T cells, have a similar onset of disease. However, the NKG2D deficient recipient develop a less severe disease than the wild type mice. So these results are supporting our hypothesis that NKG2D plays a role in the severity of the disease in the animal model, and we're interested to investigate that in humans. So the question we ask in human is this: is there any ligands that are elevated in the CNS of MS patients? So just as a recap, there's multiple ligands that have been identified in humans. So I now tested all of those ligands by Western blot using lysate from brain tissues from MS patients, but also from control. And in fact, I would like to draw your attention that the one she saw was ULBP4, and I will show you the data further, but this is the human ligand as, I would say, the greatest similarity to Meuth1, which is the only ligand we have identified as being elevated in the EAE MOG-B6 model. So Anna, by Western, saw the presence of ULBP4 in brain lysates. And when she did quantification, it became clear that the levels are higher in either active, chronic active, or even normal appearing white matter from MS patients compared to what she could measure in either gray matter or white matter from controls, or gray matter, normal appearing gray matter from MS patients. Anna subsequently tried to assess the level of ULBP4 by immunohistofluorescence, and she was able to see a greater number of cells that were ULBP4 positive in postmortem brain tissues from MS patients. And she also did co-localization with GFAP. And she saw that astrocytes are a good source of ULBP4. We could see a couple of cells that were positive for ULBP4 also in controls, but those number of cells were lower than in MS patients. And I can show you here a nice co-localization of ULBP4 and GFAP, demonstrating that astrocytes represent the predominant brain cells expressing ULBP4. And then after that, Anna wanted to investigate what kind of stress conditions could trigger such expression by human astrocytes. So she used our well-established primary culture of human astrocytes, and she exposed those astrocytes to different stress conditions: oxidative stress, ER stress, and inflammatory cytokines. And all of those conditions increase the percentage of cells expressing ULBP4 in our in vitro system, telling us that astrocytes are quite responsive to those stressors and by upregulating this ligand. She also looked into the cerebrospinal fluid of MS patients, and she observed that the band of the full-length ULBP4 was detectable in many samples coming both from MS patients but also from other neurological disease controls. However, she observed that the band of ULBP4 at a lower size, which is 25 kilodalton, corresponding to the extracellular part of ULBP4, was detectable in a subset of MS patients. Anna did the quantification in the CSF, and what she observed is that the amount of ULBP4 at the 25 kilodalton was greater in female MS patients CSF compared to either control coming from males and female donors, or also compared to the male MS patients. So we still don't know why female MS patients have those greater level of ULBP4 at the 25 kilodalton form, but this is still under investigation. So given the level of soluble ULBP4 in some of those CSF samples, Anna decided to investigate whether or not the soluble form has some, I would say, biological activity. So she took activated CD8 T cells isolated from humans, and she activated them in vitro in the absence or presence of soluble ULBP4 and assessed whether or not she could pick up any differences in the secretion of cytokines. And what she observed is that when the T cells were activated in the presence of soluble ULBP4, the amount of GM-CSF, but also interferon gamma, was significantly increased. So I would like to remind you that interferon gamma and GM-CSF are two pro-inflammatory cytokines have been implicated in the MS pathobiology. So to recap this section, we know that in MS, there's definitely disruption of the blood-brain barrier. We know that leukocytes, including T cells, CD8 T cells, can definitely infiltrate the CNS of MS patients. Another group has previously shown that most, if not all CD8 T cells that are present in postmortem brain tissue of MS are NKG2D positive. Also showed you that we can find in MS lesions, MS tissue, ULBP4 expressing astrocytes, and I show that in vitro, different cellular stress could trigger on astrocyte the expression of ULBP4. We also observed an increased level of the soluble form, the small extracellular form of ULBP4 in the cerebrospinal fluid of female MS patients, and male MS patients and control had similar levels. And our in vitro assays show that soluble ULBP4 added to activated CD8 T cells can increase their production of GM-CSF and interferon gamma. I did not have time to show you other data we have, but the soluble ULBP4 also affects the motility of CD8 T cells. So I would like to mention that it seems that ULBP4 amongst all the human NKG2D ligand has been reported not to be expressed in brain tumors, adult brain tumors. So that could be an interesting observation, because if we want to target the NKG2D pathway, if ULBP is specifically upregulated in the inflamed brain, but not in the case of brain tumors, we can think that that's a specific ligand to target in MS or even other inflammatory diseases without affecting the immunosurveillance. So this is the most important slides. I would like to thank all my current team and previous lab members. I mentioned the names of those individuals that were involved in the work I presented today. I would also like to mention the great collaboration I have with my colleagues, Alexandra and Catherine Larochelle, who are also working at the SHRM and members of your team, and also the key contribution of the MS clinic and the high quality of the samples we get is really due to the excellent clinic we have at the SHRM and of course the funding agency. So I'm going to stop here and I will be happy to answer to any of your questions.
Thank you very much, Nathalie, for these intriguing findings in two systems which are relevant for multiple sclerosis and which may be relevant to other diseases as well. Before I find questions here in the chat, I would like to start with one. You mentioned that it would be specifically beneficial to target the NKG2D signaling pathway in multiple sclerosis in order to act anti-inflammatorily. Are you aware of substances which one could use right away? Have you started with that? Is that used in other diseases?
There has been, thank you for this very relevant question. So there has been one clinical trial blocking the NKG2D receptor in another inflammatory disease. It was inflammatory bowel disease, and it was just a phase 1, but it was very promising. So definitely it was well, I would say, taken by the patients, and they saw some improvement in some of the patients. But on the long run, we think it will be better to target the ligands, because the receptor NKG2D is always expressed by multiple immune effector cells, so NK cells, NKT cells, and most of human CD8 T cells. So we think that by targeting the ligands, which are mainly, would say, not expressed by healthy cells, we can be more specific in targeting this pathway. So we currently testing something, like a similar approach in the mouse, because like I mentioned, the equivalent of ULBP4 MULT1, we also saw it as being elevated in EAE B6. So we're testing that right now, but we were worried because if we go directly just for the NKG2D pathway, it's also an important pathway in cancer. So it has been shown that is key to, because it's almost like an alarm system that goes on when abnormal cells are expressing the ligands. But so far, the literature suggests that ULBP4 is not expressed by adult brain tumors. So we think it would be a very interesting target, but definitely we want to go further. And we also currently trying to get access to other CSF, because we mainly use MS patients and also other neurological disease controls that were mainly non-inflammatory. So it would be interesting to, I would say, expand whether or not is it something that we said, I don't know, like Alzheimer, Parkinson, or even like viral infection, for example, just to see if it's inflamed, upregulated inflammation. So but definitely this is something we want to continue, because I think it would be more specific.
Okay, and only a quick question. The knockouts were full knockouts, right? Not conditional knockouts.
They were full knockout.
Full knockout.
I would like to mention this was a recent paper in JXMed published by another group showing that the specific knockout in T cells, so they use a CD4 crease. So of course, during development, CD4 is expressed by both CD4 and CD8. And they also saw like a reduced EAE in this particular model if you knock out NKG2D only on T cells. So definitely T cells were involved.
Okay, then I have a question by Sven Meuth, one of the speakers. I hope technically this works now, that Sven is posting his question.
Sorry, Frauke, I had exactly the same question, if it was conditional or full knockout, but maybe I can add after this question is already answered. I always thought that NKG2D is signaling downstream via DAP10 and DAP12. So is it maybe also a strategy to block one of these downstream molecules, given the broad expression of the receptor?
But DAP10 is also used by other, receptor, because you're right. I mean, NKG2D does not have a signaling tail, so it has to use DAP10 or DAP12. The difference between human and mice is that in humans, most of the CD8 T cells will use, most of the NKG2D is linked to DAP10. However, interestingly, there was a recent paper showing that effector memory CD8 T cells are highly activated. It seems that the NKG2D is linked to the DAP12. And that's interesting because it raises the concern that maybe when because the signaling downstream of DAP12 and DAP10 are not the same. So maybe those effector cells become even, I would say, they don't even need a TCR activation to start being toxic, because they signal through another chain. So that's really interesting. But I agree with you. Because DAP10 is also used by other receptors. So if we target DAP10, we may also affect other pathways. So that's one of the questions that we still have to, it's not simple, unfortunately. So
Nathalie, we have to be quick now. I only want to let you with one quick question and quick answer, please. Is it known, or have you looked at what cells produce IL-27?
In the brain, definitely astrocytes produce it, so they're good producers, and also myeloid cells, so microglia and macrophages.
Okay, thank you so much. And with that, we come to the next talk, to the Data Blitz talk by Alex Waldman. He is working in Oxford and the NIH, and he's talking about the prevalence and distribution of stage demyelinated lesions in the MS spinal cord. Thank you very much. Your talk.
Hello, my name is Alex Waldman, and I am a dual degree candidate in the Emory Medical Scientist Training Program and National Institutes of Health Oxford Cambridge Scholars Program, and I have no conflicts of interest. Today, I will be discussing the prevalence and distribution of stage demyelinated lesions in the multiple sclerosis spinal cord. This topic is timely given that it has been the subject of considerable debate, and understanding this fundamental aspect of neuropathology has the potential to provide insight into the etiology and pathogenesis of a major MS disease hallmark. Therefore, to address limitations of a limited number of neuropathological and radiographic studies on this topic, we performed the largest characterization of stage lesion topography in the multiple sclerosis spinal cord. We leveraged our unique cohort of 119 MS cases for which we had access to cervical, thoracic, and lumbar spinal cord tissue. Sections from each region were assessed for demyelination using proteolipid protein immunohistochemistry, and lesions were staged with the myeloid marker CD68 into active, mixed active, and inactive subtypes. The prevalence of lesional pathology was determined in the cohort, and lesions were drawn onto standardized templates before probabilistic analysis of lesion distribution was undertaken. With regards to the prevalence of demyelination, as evidenced by the pie charts here, 76.5% or 91 out of 119 cases harbored at least one lesion in the spinal cord. When looking at inflammatory activity, 87.9% or 80 out of 91 cases had evidence of lesions with inflammatory activity, staged as either active or mixed active and active. When stratifying lesion prevalence by level, we observed that demyelination was most common in the cervical spinal cord. With regards to the distribution of demyelination, hotspots included the lateral corticospinal tracts, the dorsal columns, and the gray matter as a whole. However, the most striking finding was the relative subpial sparing across the length of the spinal cord neuraxis. This pattern seems to mirror the distribution of venous drainage so elegantly described by Torben Fogh in 1950. Overall, our prevalence and distribution findings paint an interesting yet unified picture. The topography of lesional pathology in the spinal cord across all levels spares the subpial surface and mirrors the distribution of venous drainage. The cervical spinal cord is more susceptible to demyelination. Heterogeneity and mechanical stress along the length of the spinal cord was hypothesized by David Oppenheimer in 1978 to underlie this enhanced susceptibility. We take this one step further and believe that cerebrospinal fluid-derived neurotoxic factors are not the major mediators of spinal cord demyelination, but rather the prominent mobility of the cervical spinal cord transmits tensile forces onto the vasculature, which ultimately promote barrier dysfunction and myelinotoxic immune cell activation and infiltration, leading to demyelination. Thank you very much for your attention.
So thank you very much to Alex Waldman. Unfortunately, we have a bit of a technical problem to now have him answering all the questions. I would encourage you to please write your questions and you will get an answer in a written way, since these technical problems couldn't be solved so quickly. But this gives us a little bit more time at the end, and we are now really happy to have Sven Meuth from Düsseldorf talking to us. Sven is a very good colleague since years, and he is now director of the neurology clinic in Düsseldorf University. And with that, he will give the last talk, a translational talk. And his clinical translational research talk will be on non-canonical treatment targets in multiple sclerosis. Thank you very much. And Sven, please unmute, and it's your talk now.
My topic today is non-canonical, or maybe also underexplored treatment targets in multiple sclerosis. And as an introduction, I would like to mention that I have no conflict of interest. And I would like to start with the MS pathophysiology or pathogenesis. And I think neuroimmunology in general and MS in particular really developed over the last 2 decades, resulting in a number of specific compounds interfering with the pathophysiology of the disease. For instance, glatiramer acetate is a molecule where we can interact with the activation of T cells by blocking the T cell receptor MHC molecule on antigen presenting cells. With the members of the sphingosine 1-phosphate receptor inhibitors like fingolimod or siponimod, we are able to inhibit the egress of activated cells out of the lymph node. With the very specific monoclonal antibodies like ofatumumab or ocrelizumab, we are able to selectively deplete B cells, and with alemtuzumab, for instance, we are able to inhibit all cells expressing CD52, so namely T and B cells. And when we look from the periphery to the circulation and the blood-brain barrier, we are able to inhibit the transmigration of activated T cells via the blood-brain barrier by using natalizumab as monoclonal antibody. However, to directly interact with inflammation in the central nervous system, options are somehow limited. However, we have already examples like siponimod acting on S1P5 receptors with a potential to lead to remyelination. And one of the new treatment targets, so-called Bruton's tyrosine kinase inhibitors like tolebrutinib or evobrutinib, we have a dual mode of action. So we have a chance to modulate B cells in the periphery, and in parallel, we are able to interact with cells of the myeloid lineage like macrophages or microglia cells. And for tolebrutinib, it could already be shown that this small molecule reaches the central nervous system. However, when we talk about non-canonical or underexplored treatment targets, we have a whole bunch of different topics. And due to time reasons, I was able to select a few number as pars pro toto. And one example is modulating organs like the skin or the gut by environmental factors. So the modulation of environmental factors or lifestyle factors might open up a new strategy to complement treatment strategies we already have. And one example here is that we modulate the immune system of the skin, which results in a modulation of the immune cell phenotype in lymph nodes, and consecutively, therefore, we might impact inflammation in different other organs, for instance, the central nervous system. And one example is maybe UV light, because we know that this UV light is able to modulate the vitamin D metabolism. And this, for instance, in the skin leads to the upregulation of rankle, which is accompanied by an additional expansion of T regulatory cells, leading to systemic immunosuppression. And I think one nice example was presented by Ostkamp and colleagues last year, published in PNAS, where the group could show that the latitude and therefore the sun exposure of patients is correlated with the disease progression, for instance, the expanded disability status. However, in the very same work, a group of patients was treated with narrowband UVB therapy, and after cell sorting, RNA sequencing, sequencing and bioinformatic analysis, the group could show that there are transcriptional changes in the different immune cell subsets like CD4, CD8 T cells. And this is, in my opinion, one prime example where we have a chance to additionally modulate the immune system by modulating an environmental factor or a lifestyle factor. And along the same lines, I think we have to discuss about the role of the microbiome. And therefore, I selected a Cell paper from 2020, presented by Dusha and colleagues, and they could show that a short-chain fatty acid, namely propionic acid, is able to induce Treg genes in the gut. And this results in the immune compartment to an increase in regulatory T cell numbers, an increase in the metabolism and function of regulatory T cells, for instance, the production of IL-10, and finally, a suppression of pro-inflammatory Th17 positive cells. And as you can see, within this same paper, the group investigated approximately 100 MS patients treated with propionic acid, and there was a significant reduction in the annualized relapse rate, and the number of patients with a confirmed disability progression, disease progression, was significantly lower in patients treated with propionic acid compared to non-treated patients. And this resulted finally also in a decrease of the EDSS comparing the time after a PA treatment with the time before patients were treated. So this is another, in my opinion, prime example where lifestyle factors like nutrition might have a relevant impact on systemic immunomodulation. And a second topic, and this is something my group is also very much involved in, is the potential of modulating ion channels during the disease course of multiple sclerosis. And I think one interesting ion channel family we are investigating are so-called K2P channels. And as shown in this cartoon, these are channels with two subunits forming a pore, which then liberates potassium out of the cells. And over the years, we were able to show that these ion channels are important for immune cell functions, several members of the family, but they are also expressed and they are regulating the activity of neurons and neuronal networks. And one candidate, TREC1 channels, they could be shown to modulate the blood-brain barrier in a way that they regulate the transmigration of immune cells via the blood-brain barrier. And this candidate here, TRESC channels, they are described to be mainly expressed in the spinal cord, but we could show that they are also very important on different immune cell subsets and preferentially in regulatory T cells. And this is a paper published very recently by Tobias Ruck and Stefanie Bock from our group. And they treated EAE mice with a channel opener, cloxyquine. And what you can see here is the typical EAE disease course in untreated or sham-treated wild-type animals and in cloxyquine-treated animals. And you can see the amelioration of the disease course. And this is accompanied by an increased frequency of CD4 positive, CD25 positive, FOXP3 positive regulatory T cells in the brain and in the thymus. And interestingly enough, this data could be translated in the human situation because we have humans with a wild-type channel and we have humans with a amino acid exchange at position 110, and this leads to a non-functioning of the ion channel. So we can ask the question in the human situation, what does it mean if this stress channel is not functional in our patients? And what you can see, and again, with cloxyquine, we activate the channel. Here, we block the channel in humans. And you can see that the frequency of regulatory T cells is significantly decreased in the patients carrying the SNP. And this is also true for recent thymic emigrant Tregs. And then we asked the question, can we use this mechanism maybe also for a treatment option for our patients? So what happens if we activate the channels in the human situation? And luckily enough, there is one antibiotic, so-called nitroxalin, which is able to significantly increase the activity of the channel. And when we treat patients with an urinary tract infection with this antibiotic, over 7 days, you can see a significant upregulation of regulatory T cells over time with up to 40% increase in the frequency of these regulatory T cells. And this is a transient effect over time. And when we compare it to another antibiotic, you can see the specificity of this treatment. And maybe as a final result, I would like to show the mechanism. So after T cell receptor activation in CD4 positive T cells in the thymus, we have an initial calcium signal leading to the upregulation of transcription factors like NF-kappa B, and this results in an overexpression of the channel. The channel, by mediating potassium to the outside, reduces the voltage, the membrane potential, leading to an even higher calcium signal, and this finally leads to the expression of FOXP3 regulated genes, namely the channel mediates the existence of regulatory T cells. And as another example for ion channels, I would like to mention TRAC1 channels because they could be shown in a Nature Medicine paper some years ago to regulate T cell transmigration via the blood-brain barrier. However, in these days, the exact mechanism was not clear, and very recent data can show that obviously endothelial cells build up protrusions upon inflammation, here shown in wild-type endothelial cells treated with TNF-alpha and interferon gamma. Compared to this situation, atomic force microscopy revealed that these protrusions are already present in the TRAC-deficient mice, and this is also shown in these two photon images here, where we can show as soon as we block the channel, the T cells are able to transmigrate via the blood-brain barrier. And the mechanism behind seems to be a reactivation of the actin cytoskeleton, so a increase or decrease of these protrusions. And using confocal microscopy, we were able to show that ICAM is expressed on these protrusions. So TRAC1 leads to the formation of, let's call it, kind of sticky fingers. And maybe as a last example, I think an overall goal in the treatment of autoimmune disorders is to selectively suppress effector T cells without inducing a general immunosuppression in these patients. And there are several strategies to induce immune tolerance, for instance, antigen-specific tolerization, or as shown here, using tolerogenic professional antigen-presenting cells, or antigen-unspecific tolerization, like extracting CD4 T cells from patients, then ex vivo expansion and differentiation to regulatory T cells, and then one can use the regulatory T cells in the very same patients. However, there was a very recent science paper showing that mRNA vaccination could also be used to induce this tolerization in a very specific way. And here people used a nanoparticle formulated mRNA encoding for proteins relevant to the autoimmune disease. And in this case, in the AE model, they used MOC 35 to 55, so myelin oligodendrocyte glycoprotein, and they could show that they nearly prevent the existence of the disease compared to an irrelevant mRNA or mRNA encoding an irrelevant protein or control values. And this was accompanied by a significant reduction of CD4 positive T cells, for instance, in the spinal cord. And as a proof of concept that this is not only MOC specific, but in a general principle, if you use relevant peptides or proteins, they could show that it's working for MOC, but also for PLP, another peptide expressed on oligodendrocytes. And again, when they looked in the brain after the vaccination strategy, they could show that, for instance, pro-inflammatory IL-17A positive T cells are significantly reduced compared to controls. And therefore, I think we have still room for new treatment strategies. And I would like to thank you very much for giving me the opportunity. And again, due to time reasons, I could only present a selection of strategies, and I will be very happy to take your questions. Thank you.
Yeah, thank you, Sven, for this wonderful, inspiring talk with all these great examples. I encourage the audience to ask questions. I don't have them yet in my chat, but I'm coming to one first question. Sven, while the vaccination strategy to induce regulatory T cells seems to be still antigen specific, all these molecules are myelin molecules, no others have been shown. Your strategy to foster Treg response seems to be a little bit broader, maybe more easily applicable. So how are the next steps concerning treatment in this chapter?
Thank you for this question. Indeed, the strategy to open up these stress channels lead to an increase of regulatory T cells from approximately 2 to 3% in the peripheral blood to up to 9%. And this obviously somehow inhibits inflammation. I have to say, when we had this translational approach to bring this into patients using this antibiotic, we were happy to see that on this short course, you get an increase in the regulatory T cells. However, normally the antibiotics are only given for 10 days. And actually, I think the next step should be something like a phase 1, to really ask the question, are we able to give this antibiotic for a longer period of time? Or if we do need, let's say, kind of pulses treatment courses to come up with a higher number of regulatory T cells. And on the other hand, we all might remember that there was a strategy to significantly increase regulatory T cells by anti-CD28 antibodies. And we all know that this resulted in a cytokine storm and with significant side effects in these healthy volunteers. And so I think it is a strategy, and we are very happy to follow up on this, but we also see a certain risk.
Okay. Okay, good, Sven. This was a wonderful ending point since we ran out of time. You should please answer several questions which may follow via the chat and via the platform. And for now, I thank the audience. I thank all discussants, all speakers, and certainly specifically the last speaker on this translational avenue in multiple sclerosis. I hope this was inspiring for all of you who are in the audience. Thank you very much and have a wonderful ongoing Congress.